Okay, so what we had talked about last time was semi-conservative replication, and this is where a new double helix has one conserved old strand and a new strand. So what we're going to talk about now is what specifically is happening when DNA is replicating. So here is bird's eye view of DNA replication, and as you can see here, we have our template strand that's going to be here in black, and then we have our hydrogen bonds, and then we have our other template strand, which is located here. So again, I want to remind you, when we're talking about DNA replication, you always want to be mindful of the orientation of DNA. So here we have our 3' to 5', and then we have our 5' to 3'. And that's important to know because when we talk about how the enzymes come into play, we need to know the orientation of the DNA. So first we have our double-stranded DNA. So start with the double-stranded DNA, and that's what we can see here. 3' to 5', 5' to 3'. And in between those are going to be our hydrogen bonds. Then, as DNA is beginning to replicate, the origin of replication starts. This is also called the replication bubble. You can see here at the center, you have this sort of bubble formation coming up, and that's where the DNA is beginning to replicate. So then, as we're looking at the orientation, you see for the top strands can be 3' to 5', but here at the bubble, we're going to have our new strands of DNA come in, which is going to be these pink ones. And notice that if we're looking at a 3' to 5', here the opposite strands being made. So here you can see a 5' to 3'. So on this side of the replication bubble, this is going to be fork 1. And at the end of this process, we have 2 strands of DNA being made. So again, let's go back to part 3. It says 2 replication forks form and move in opposite directions. So it's fork 1, fork 2. And then it says at the end, 2 identical copies of double-stranded DNA are formed. So here we have our 1 strand of DNA that came from a template there, and then we have our other one here. So here are 2 identical copies of double-stranded DNA.